the evolution of Hoxa11 gene, and why we have 5 fingers. I'm Callie Valtierra.
I'm Shivani Bhagat.
I'm Hoxa11. And I'm Brianna Hayden.
In a world of millions of possibilities, why do we only have 5 fingers? What prevents us from having 6, 7, or even 100? Through years of evolution, we have ended up with the magic number 5. But what allows it to be that way? Genes. No, not those genes, these genes. We'll explain how gene regulation works to ensure that we have our 5 fingers. General definitions. Homeobox genes. This is another name for Hox genes. Homeobox genes are important regulators for the growth of limbs. Pentadactyl. This structure includes animals which have 5 digits on an extremity. For example, humans have 5 fingers on a hand and 5 toes on a foot. This structure makes up amphibians, mammals, reptiles, and birds, meaning that these species share a common ancestor. Polydactyl. This type of structure includes animals which have more than the normal number of fingers or toes.
Metazoan. These are the animals which go through development from the embryo stage with 3 tissue layers. Autopod. This is the most distal region of the body which contains the hand or feet and the digits which they are comprised of. Tetrapod. A tetrapod is a vertebrate possessing 2 pairs of limbs or a total of 4 limbs, such as amphibians, reptiles, birds, and mammals.
Background and origin. The Hox gene first originated from the proto-Hox. The proto-Hox is a specific gene cluster which existed before Cnidarians, dating to be 580 million years old. This means that the proto-Hox gene cluster came before the formation of vertebrates. The proto-Hox cluster had approximately 2 genes, and through cis duplication, or the duplication of genes on the same chromosome, the formation of Hox genes and para-Hox genes began. The Hox gene. The Hox gene, which formed after the cis duplication of the proto-Hox gene, makes up many of the animals seen today. The Hox genes are a set of transcription factor genes that give a glimpse of how gene expression is translated into the formation of metazoans. The genes are present in the genomes of all animals, including plants and fungi. Mutations which occur on the Hox gene tend to have 3 different outcomes of the replicated or copied Hox genes. The mutations alter how an organism looks.
The different Hox genes include Hoxa11, Hoxa13, and Hoxd13. The Hoxa11 gene is a protein-coding gene which provides cells with specific positional identities on the anterior-posterior axis. The gene allows people to have their limbs in the places they need to be. The Hoxa11 gene is believed to have emerged mainly in polydactylous structures. Regulation of this gene led to the transition from polydactylous structures to pentadactylous structures. This gene is activated by the Hoxa13 and Hoxd13 genes. The Hoxa13 gene provides instructions for producing a protein that binds to specific regions of DNA and regulates the activity of other genes. It deals with the formation and development of limbs, the urinary tract, and the reproductive system of the embryo. The Hoxd13 gene is located on a cluster found on chromosome 2. The removal of this gene cluster can lead to severe limb and genital abnormalities.
Expression of Hoxa11 and Hoxa13 genes. It shouldn't surprise you that the pendactyl features of humans and mice originate from the fish fins. According to research done by Dr. Kamita from the University of Montreal, the expression of fins and fingers are both due to the Hoxa11 and Hoxa13 genes. In their research, they discovered that the major differences between fins and fingers is the arrangement of the genes. In fish, the Hoxa11 and Hoxa13 overlap and fins are produced. In humans and mice, the genes remain separate to produce a pendactyl state. In the experiment, however, fish genes were inserted into mice embryos, and mice fingers mutated to produce 7 digits. Why did this occur? Well, let's step back and take a deeper look into Dr. Kamita's research.
The graph featured here displays the activation of the Hoxa13 gene and its effect on GFP, which stands for green fluorescent protein. GFP is a measure of gene expression. Quite clearly, the graph displays how there is more expression when the Hoxa13 gene is on.
Why does this happen? Antisense transcription of the Hoxa11 gene is why this happens. What is antisense transcription? Antisense transcription is a form of gene regulation. As we know, transcription, the copying of a DNA strand into mRNA. This mRNA is later translated into a protein which serves as a way to express the gene. Through the antisense transcription of the Hoxa11 gene, the antisense mRNA created is inhibited from translation into a protein. In fact, by base pairing it, the translation machinery is physically corrupted. Thus, the Hoxa11 gene serves as a regulator and is not expressed. Only the Hoxa13 is truly expressed, which leads to a pentadactyl state as opposed to a 7-finger state.
Even though our project was centered on the Hoxa11 gene, which regulates the formation of our 5 fingers, the Hox gene as a whole helps normal limb development during the embryonic stage. Next time you see someone with 6 fingers, you won't have to ask why.